Hey, and we are back, and we're going to start a new series today, and we're going to jump around. We're going to come into the biohacking space, and we're going to go big. We're going to go straight off with gene editing technologies. Yeah. So, recent paper, recent advances in therapeutic gene editing technologies, June 2025. Yeah. In the Journal of Molecular Therapy, which is a pretty big one well, specifically to this area, one of the biggest ones. But yeah, so big recent review paper. So, and we've got three authors, Dongqi Liu, Di Cao, Ren Zehan. Check out my Mandarin tones. Tones, meh. Hopefully, at least the pinyin is the characters are pronounced vaguely correctly. Although that "j" one is always that's a nightmare, right? Anyway, okay. So, recent advances in therapeutic gene editing technologies. Yeah, it's starting now for health. Yeah, but once you can edit your genome, we'll see. Yeah, we'll talk about that later. Yeah, there's all sorts of fun stuff. Let's jump straight into the abstract. The advent of gene editing technologies, particularly CRISPR based systems, and CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And we will come back and explain that in detail later. It's been in the news a little bit now. It's our main technique. We nicked it from bacteria. And I'll explain in detail how it works. But basically, it means that you can insert stuff into DNA. Yeah, snip it open and stick stuff in. It's very cool. So it's a biologically inspired gene editing technology, like all the good stuff is, man. Biologically inspired computing, biologically inspired biology. Yeah. So CRISPR-based systems has revolutionized the landscape of biomedical research and gene therapy. We'll touch on the research stuff. Obviously, that's the main, it's a big application, and gene therapy for disease. But then we will sci-fi it up. Yeah. Ongoing research in gene editing has led to the rapid iteration of CRISPR technologies. They're blowing up now. Yeah. So we've got base and prime editors, enabling precise nucleotide changes without the need for generating harmful double-strand breaks. DSBs. We don't want those DSBs, right? We want to just snip it once. Yeah, minimum amount of snipping. It's like surgery. Similar kind of system, right? The less invasive you can make the surgery, the less chance of bad complications. The less choppy you get with somebody's DNA, right? The better it is, right? The easier it is to make sure that the error rate is low and it snips back in the right place and everything, right? Furthermore, innovations such as CRISPR fusion systems with DNA recombinases, DNA polymerases, and DNA ligases. Right, so I have to make sure I get all these ones right. But it's basically opening, joining together, and then sort of recombining is another type of joining together. So we'll go in detail later. We're just on the abstract, but all the way you got all these, and these are all enzymes. So ases means enzymes, right? So these are enzymes that work specifically on DNA, and they're either cutting them, squishing them together, or merging something in and then squishing it together, right? So on top of the fact that we have a specific thing for inserting and finding the locations where you need to do, because that's the tricky thing, is like finding where you need to cut and where you need to insert, right? We got all sorts of other things for repairing it and fixing it afterwards now, as well. Yeah. So we're starting to build the DNA working tool set. Yeah. Have expanded the size limitation for edited sequences, opening up new avenues for therapeutic development. Beyond the CRISPR system, so there's also other stuff, right? We've got mobile genetic elements, and we'll dive into those later. I'm not an expert on that. I'm not an expert in any of this area, by the way. Mathematical ecologist, right? So, but like I did, in my undergrad and masters, I've done genetics, right? Masters Ecology, Evolution and Conservation. So there's quite a lot of genetics modules in there. So, you know, I can talk the talk, but I've never worked in this field. I've never published anything to do with any of this stuff. Epigenetic editors are emerging as efficient alternatives for precise large insertions or stable gene manipulation in mammalian cells. Yeah, epigenetic editors that's cool, right? So that means that you can. That's like live genetic editing stuff, yeah. So, well, epigenetic means that you're looking at further on systems. So, like my classic example of epigenetic effects would be something like a maternal effect gene. So it's a genetic effect, but it's in the mother. The DNA lies outside of the baby, right? But it's going to affect the development of the child, right? So that's not just because sometimes people get this phrase wrong. This phrase is really in sort of science communication and sort of mainstream. Everyone's like, oh, epigenetic, whoa. And they're just talking about phenotype, right? They're just talking about the expression of genes through the environment. That is not epigenetics, yeah? Epigenetics is not just like if you give somebody more food when they're growing, they get taller. That's just phenotypic expression. That's just the interaction of genes and environment. Yeah, which leads to genes, genotype, genes plus environment, phenotype, right? So that's not epigenetics. Epigenetics is systems that that have impacts on the way that your genes are expressed that sit outside of your immediate genetic environment. So this might include things like protein folding, right? So this is like you've got non-nuclear elements like histones in your DNA that are going to change the way that your DNA is like able to fold around. So that means that things aren't going to read or they're going to selectively read it at certain times. So you can change gene expression through non-genetic things that could be conserved. Yeah. So, like the structure of these protective proteins like histone proteins and other things like junk DNA that we didn't think was being coded, but actually is doing that same sort of structural function. So it's not junk, right? Terrible name. That's a terrible name. I hate that name. But anyway, so that's one of my little pet peeves. Yeah, people just being like, that's everything. Everything's epigenetic now, and also because I do this sort of, I had an Instagram thing called Gym Rants, and there's a lot of kind of bro scientists in fitness. Yo man, my genetics are bad. That's why I can't. They use all the wrong words, and the epigenetics started appearing in some guys' feeds, and I was just like, that's not what that is, all right? Anyway, people use the wrong words for things in fitness. I'm sure I'll do one about that at some point as well. Yeah, maybe talk to one of Luke's stories. then we can go deep on biohacking for health and fitness. Yeah, and gym bros and gym culture. Right. Yes. So. Yeah, epigenetic stuff is cool because as far as I know, and I'm not up to date, so maybe I'll pick some other stuff. A lot of these we don't understand. Like, we don't understand the folding stuff. But that is also an area, it's one of the few areas where I think that it's totally cool to use AI, right? Because we are rubbish at this. This is too complicated a problem for human beings to see the patterns in. There are a few applications where I'm like, well, we just literally can't do this. This isn't stealing a job from anyone. This isn't, replacing human flourishing. It's not automating pure mathematics to put maths professors out of a job. Get good maths professors, retrain as a plumber. Come on, get with the modern economy. What? Why are we putting effort into that? Math professors are perfectly good at doing maths, right? It's good for them, and they're not going to do other stuff, right? You've seen those guys, yeah. And for me, that is like a high art as well. True, pure mathematical research is these guys, the guy that did the Andy something, I've forgotten his name, but he did the the paradox. Oh man, I'm gonna have to look it up now. Okay, Andy, maths researcher, paradox. Oh man, I'm gonna. Andy Wiles, yes, yeah. Not it's Fermat's last theorem. Yeah, and Andrew Wiles, Fermat's last theorem. Like, this guy is like Michelangelo, right? His story is insane. He's just laboring away, and then he's forced into go and doing some teaching, and he just teaches his theorem, and there's only about. There's like 4 PhD students that come to the lecture, and like the 2 other professors that work in his area. And by the end of lecture 3, there's just 1 professor left, and like no one else in the world could follow his work. So like it. But eventually it was proven to be correct, right? And his absolute dedication to this, unified 2 areas of science and brought up the skill level and the ability of humanity, right? Which is awesome, right? He's the Michelangelo of maths of our generation, right? Like, he's just pure genius and passion, right? Laboring for years. And everyone says he's insane, he's delusional, right? And then he brings this thing into the world, and then he's unified 2 completely disparate areas of mathematics, proven something that people thought was unprovable for 100 years, and now people are writing books and studying his work. The point of doing this stuff is to improve human understanding. If you just automate it out to machines, it's a complete waste of we don't know it. It's the death of knowledge, right? It's this is XT276 has formalized step 473 and corrected the lemma on page 48 at the same time. All right. Yeah, if you say so, mate. Like, anyone following any of that? Well, X7643 checked his work, and we assume it's doing a pretty good job. All right, anyone understand the output there? Yeah, it doesn't matter. It's all optimized. Like, we checked it via debate, and we know that they're roughly making the right kind of arguments. So you're just judges and jury now. You're not mathematicians anymore, and you don't understand anything that's going to lead to human flourishing. Well, when you put it like that, it seems a little bit strange, doesn't it? yeah, it fucking does. Right? So that's my opinion of these kind of technologies. Yeah, like for a lot of things. So AI is just completely awful, right? For a lot of things, right? It's just going to destroy. It's going to lead to the death of knowledge, right? It's not going to set you free. It's going to mean that things are happening we don't understand, right? Even the best and brightest minds in the world won't understand it, right? Like, what's the point? Why do we want those developments if we don't understand any of them? Like, we will stop standing on the shoulders of giants, right? And we will start being underneath a deck of cards, an infinitely tall deck of cards being built by robots that are just designed to build more cards and stack them on top of you forever, right? And then when they come crashing down, we're like, well, I didn't really understand the 6th dimensional architecture of the tower, as it crushes everyone in humanity to death. Maybe that metaphor got away with me. But anyway, but there are a few specific applications that don't do that, right? Because there is no way for us to do this. Like, incredibly complicated systems like decoding whale song and protein folding and epigenetic histone folding and how the DNA fits together in those complicated shapes. Like, I think it's okay to like hand in the card and say, like, yeah, we don't understand the structures there, but like, they're so important and useful that we might be able to extract something from it. Nobody's losing a job. Nobody's losing any sleep over it. No one's got any, linguists tried all the whale song stuff for years. It's like, we ain't got that shit, man. Yeah. And then there's the potential to maybe analyze the structural elements of those computations afterwards, and then see, find the pattern within the pattern, right? And so that it can be used to support human knowledge and understanding, not just train us out, right? So it's not only the way that the application is, it's also it's not only the nature of what it is being applied for, but it's also how you go about doing it. Like, both of those two things need to line up so that your AI is in alignment with human flourishing, rather than just like automating us out of existence. Yeah, which, because like who is that in service of? Like, it's in service for a few rich people that are going to own those, right? And like, and also the shareholders of Nvidia, you know, like, and it's not in service of mathematics. It's not in service of human knowledge. It's not in service of human flourishing, and it's not in and therefore also mathematics. You know, those abilities undermine physics, chemistry, but so like again, who's benefiting from this? Yeah. A few rich people for now, yeah, until all of their jobs are automated out as well, right? Yeah, like just be careful with this stuff. But coding epigenetic elements and then scientists studying that to learn the underlying patterns when they couldn't see it before, Mark approves. Yeah, like that's okay. That's because that to me is because I also see other applications like where that to me feels like having an AI assistant in your truck driving window who's giving you AR support, right? And is reading all your biomedical signs for tiredness and all that kind of thing, and is highlighting stuff on the road and to identify when your next rest stop is and all that kind of thing. Because that's supporting the human. So the human keeps their job. They get better at their job. Yeah, they get way better at their job, right? And they're and you can test it with safety profiles and everything like that. And especially then if you sort of build a like a chat companion type thing into it as well, is it's relatable and stuff. It's also, he's got Buddy in with him in the thing, so you've increased human achievement and flourishing, right? So those lonely car rides by himself, he's now got an AI assistant companion that helps him on the road, right? Like. Or you have an automated truck, yeah, that puts millions and millions of men around the world out of work overnight, right? And decreases human flourishing on a grand scale, and increases efficiency and capitalization. And also that's the other. Also, all the legal and ethical ramifications about what happens when they start hitting people. When the first time it runs over a kid, yeah. Those problems are averted with because the system is not in charge. He's the helper, right? So it's still the human that takes responsibility. So yeah, like all of those probably, you've got one system that just has huge, tremendous human flourishing advantages, right? And also gives us better road safety and better working environments. And you've got another one that's just awful, right? But people don't seem to see the magnitude of the gap between, because they're just like, that's got AI in it, that's got AI in it. But these are two different worlds you're building, like radically different worlds. Yeah. And I think that stuff matters. Anyway, that's a tangent. Okay. But yeah, there's some cool applications of AI in this area as well. Okay. Specifically to the epigenetic protein and DNA folding stuff. Let's wrap up the abstract. These advances collectively set the stage for next generation gene therapy development, which is cool. Like we're gonna go. I'm sort of like prepping. I keep saying we're gonna go big. We're gonna go sci-fi on this. But yeah, let's just bide your time, okay, folks. Yeah, we will though. We will get to it. Yeah, we will get to improving human functions. We will get to the controversies of ending certain types of conditions. Whether we think that that's cool or not, and potentially we've got the Gattaca kind of everyone's good at everything kind of. So we've got, not just when we remove disease, then we start removing variation from the genome. That's all scary. But I think the even cooler stuff, let's just cat girls, right? Okay, cat people, maybe dog boys, panther boys, yeah, whatever, whatever you're into, we could do that. Like we already put genes from other species into plants, right? We got some fish genes in tomatoes. We got all sorts of other stuff going on, genetic editing. The first person who wants to take the jump and start messing with the cat genes. brave. Let's just put it that way. The early advances in the technology, yeah. Um, Explores the preclinical innovations poised to advance the field. So, and then we'll also talk about the last set of innovations set to get things moving. Yeah, boom. We'll be back next time. We'll look at a nice little, it's got a little graphical abstract that talks about all the different key areas. We'll jump right in with the introduction next time, and then we'll save the cat girls for later. Yeah, see you all very soon.